Hello everyone and welcome back to another video. In today's episode, we're going to be discussing a cell biology topic, meiotic division. So we're going to go over the phases in meiosis I and in meiosis II. So I hope you stick around and enjoy the video. Let's get right into it. So with meiosis, the first thing you want to do is just differentiate it from mitosis. So what makes it different than mitosis? Well, mitosis occurs in just somatic tissue cells, and mitosis results in two identical daughter cells. Mitosis goes from 2N to 2N, meaning it goes from a diploid cell, it replicates its DNA, and then it divides again, so it's back to 2N. However, meiosis is not the same. Meiosis goes from 2N to N. And how we do that is going to be described in this video. So meiosis occurs in gametocytes, which are germ cells. And it results in up to 4 non-identical sex cells, which are gametes. With regards to the difference between mitosis and meiosis, the whole point that you need to understand is that mitosis produces identical cells. Meiosis produces non-identical cells on purpose. It wants to differentiate the cells, and it wants to provide 4 new cells with unique combinations of genetic information. So the overall process of meiosis looks something like this. Meiosis I is outlined in blue. Under Prophase I, I have the 5 different stages of Prophase I. We're not going to go into that much detail, but I put them there just for the sake of completeness. And then in green, we have meiosis II. So meiosis I results in homologous chromosomes being separated, generating haploid daughter cells. This stage is known as reductional division. So notice I have 4N. This is after interphase, specifically S phase, when the genetic material has been duplicated. So 2N goes to 4N, and that's where we're at. Meiosis II is similar to mitosis in that it results in the separation of sister chromatids without a change in ploidy, and therefore it's known as equational division. Alright, so let's begin. The main point you need to get from interphase is that after S phase, with meiosis, there are 92 chromatids organized into 46 chromosomes, which are organized into 23 homologous pairs. So with interphase, we see that the nucleus is still intact, and DNA exists in chromatin form. To be more specific, it is euchromatin. So before we go any further, one note that I'd like to share with you is that if you'd like to take a piece of paper and write down what I have as bullet points, I'm going to summarize them even further, but the bullet points are there for you to read while I'm speaking and talking over the video. But of course, you can take paper and write them all down. What you need to get out of Prophase I is that the chromatin condenses into chromosomes, the spindle apparatus forms, and the nucleoli and nuclear membrane disappear. And this is where the first major difference between meiosis and mitosis occurs, because homologous chromosomes and meiosis come together and intertwine in a process known as synapsis. And this is where crossing over occurs. So understand that at Prophase I, crossing over occurs. So moving along, metaphase I, what you need to get out of metaphase I is that homologous pairs, or tetrads, align at the metaphase plate, the cell equator, and each pair attaches to a separate spindle fiber by its kinetochore. Note the difference here between mitosis and meiosis. In mitosis, each chromosome is lined up on the metaphase plate and is attached to 2 spindle fibers, which is 1 from each pole. However, in meiosis, homologous chromosomes are lined up across from each other at the metaphase plate and are held by 1 spindle fiber. During metaphase I, the important process to understand, independent assortment occurs. Moving into anaphase I, The cohesin proteins are degraded between sister chromatids, and each dyad is pulled toward each pole of the dividing cell. This is disjunction. You may hear the word non-disjunction, and non-disjunction is not a good thing. That's when, say, this one would stay attached, and it would go with this, in summary. So we see here at anaphase that disjunction accounts for Mendel's law, first law specifically of segregation. And then the last process of meiosis 1 is telophase. During telophase, a nuclear membrane forms around each new nucleus, and at this point each chromosome still consists of 2 sister chromatids joined at the centromere. We see the cytoplasm is beginning to disperse towards each pole of the cell with its genetic makeup. So reductional division has been complete. Now let's move into meiosis 2. With meiosis 2, it is very similar to mitosis in that sister chromatids, rather than homologs, are separated from each other. So beginning with Prophase II, the nuclear membrane around the dyads disappears, chromatin condenses, and the centrioles migrate to opposite poles while the spindle apparatus forms. Spindle apparatus obviously functions in metaphase. So with metaphase 2, what you need to get out of it is that the chromosomes line up on the metaphase plate. You can call it the metaphase 2 plate for simplicity. The Shugoshin complex is degraded, and the cohesin holding the centromeres together is now available for degradation. And I didn't state this in meiosis 1, but this is an important point to take note of, is that the cohesin proteins holding the centromeres together is degraded by separase, which is a pretty easy enzyme to memorize. Continuing along, we have Anaphase II. In Anaphase II, we see the same thing as Anaphase 1, except it's double the amount. And so we have all 4 beginning to form, as you can see under the microscopic image. And what you need to know for Anaphase 2 is that the centromeres are dividing, separating the chromosomes into sister chromatids. And then these sister chromatids are pulled towards opposite poles via the spindle fibers. So what we're seeing here is N, 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 and N beginning to form. Another thing to keep in mind is that interpolar microtubules are elongating the cell. And to conclude with telophase 2, chromosomes have reached the opposite poles of the cell and the distinct nuclei have been forming. What we have now is that each chromosome is a monad, or a monad, however you like to pronounce it, and cytokinesis has been initiated, which is the cleavage furrow pinching the cell, resulting in 4 haploid gametes. So I hope you enjoyed the video. I hope it summarized meiosis for you a little bit better than it was before the video. And we're thinking about making a video summarizing what occurs actually during anaphase, when cohesin proteins are broken down at the kinetic core or the centromere. And so if you'd like that, leave a comment down below, or with any other video ideas that you have in mind. Hit the like button, and we'll see you in the next video. Thank you for watching.